Well, hello everyone. Thank you really very much for being here today and for inviting me to be part of this wonderful seminar series about biosensors. I'm really excited to present some of my work that I conducted at the Joint Bioenergy Institute, increasing the production of this jet fuel precursor, isoprenol, in Pseudomonas putida. So we achieved this by leveraging and characterizing the short chain alcohol signaling cascade of this microbe, Pseudomonas putida, which is a soil bacteria that has really the potential to become a powerhouse in industrial biotechnology. And at JB, at the Joint Bioenergy Institute, our mission is to be able to revalorize plant waste biomass by using that as feed to produce bioproducts and biofuels. And we achieve that by the different divisions that form JB, that we all work as specialized engines towards the same goal. And as, for example, we are starting from the feedstock division that they work on engineering plants so that they are more resilient and they are also easier to deconstruct. The deconstruction team, which they work in depolymerizing these components like this cellulose, hemicellulose, and lignin into more bioavailable monomers like sugars and other aromatic monomers. And then in the biofuels and bioproduct division, we engineer a plethora of microbial hosts so that we can maximize the conversion of these carbon streams to different valuable products, always aiming to hitting tighter rates and yields that are important for any industrial processes. And of course, we also have a technology division, which is not clearly shown in this pipeline, but they are crucial for our work since they provide these high throughput platforms that accelerate our research. And I work in the M group, Andula mucopodice group, which operates within this biofuels and bioproduct division. And as Megan was saying before, our aim is to be able to develop and implement different methods to maximize the use of these microbes and also to be able to understand the underlying physiology of these platforms when used for these heterologous productions. And this is just a photo of our research group. And well, you might be thinking, like, what's isoprenol? Why isoprenol? So as I was saying before, isoprenol or 3-methyl-3-butanol, it's a platform commodity chemical with really attractive properties as a diesel fuel blend stock, but also because it is the precursor of the MCO or the methyl cyclooctane, which has really good combustion properties that surpass those of the current or traditional jet fuel, which is Jet A. And while looking into how can we implement this in a microbial host, we decided to use Pseudomonas putida for several reasons, and some of them being first that we have extensive expertise in the use of this microbe, and that we have many tools for genetic and metabolic engineering to do so. Then this microbe, it is highly tractable. And also something quite important is that isoprenol is an alcohol, which it has this inherent nature of inhibiting microbial growth, but Pseudomonas is particularly tolerant to this type of compounds. And lastly, and also quite importantly, is that Pseudomonas putida it has a really versatile metabolism, and it can consume most of these monomers obtained from the deconstruction of plant biomass, which is great for our pipeline. And while gas chromatography, it is really the gold standard for quantifying isoprenol in the lab, our high throughputness is really hindered or really limited by the extensive run times of the instrument and also the laborious sample preparation. So we decided to look a little bit more closely into Pseudomonas putida because, in fact, this microbe is able to consume our target compound, which in general is not desired, of course, because we aim to produce this compound. But then we were thinking that if this Pseudomonas is actually able to consume it, it means that it's able to sense it. So maybe we could harness that ability of sensing isoprenol to improve our high throughputness and designing a biosensor. And in this case, and throughout this talk, there's many definitions of biosensors, as you all are aware of. In our case, we work with transcription-based biosensors, meaning that isoprene is the inducer molecule that drives the expression of the downstream genes, natively these catabolic genes. And for our purposes, we replace that by a fluorescence reporter. And in our case, we are using mCherry throughout the whole presentation. And while the catabolic pathway of isoprene is not known in Pseudomonas putida, there was a previous publication from Thompson et al. That you can see here in this left side of the slide, in which they observed that the transposon inactivation of the gene 2675, shown in green here in this growth curve, prevented the growth of Pseudomonas putida on isoprene as the sole carbon source. This 2675 is not a catabolic gene. It participates in the regeneration of the PQQ cofactor, which is a cofactor needed for most of the alcohol dehydrogenases in Pseudomonas putida, which Pseudomonas putida has at least 14 annotated alcohol dehydrogenases, so it's complicated to actually determine if there's a single one that is actually carrying the function. So just deleting this PQQ cofactor, that's the work of preventing the growth of Pseudomonas. And I also wanted to highlight the work of Adam Deutschbauer and Morgan Price generating this RBT-seq fitness pipeline and publishing the data in their fitness browser. This is all publicly available data. And what in a few lines, just to give you a summary of what this method does, is that a library of barcoded transposons is transformed in your desired strain. So in our case, Pseudomonas putida. And then this library is grown on specific conditions. And the barcodes are sequenced at the beginning and at the end of the cultivations. And then the abundance of those barcodes at the end of the cultivation is used to calculate a fitness score, meaning that if there's a specific gene or an activation of a gene that has a lower abundance, it would result in a negative fitness value, meaning that that gene was actually important for the growth of a strain on that condition, and that's why its abundance decreased over time. And this is the pipeline that we designed and that we implemented and validated to design biosensors. As I was saying before, first we analyzed this RBT-seq fitness data, which is publicly available. And something to note is that usually when looking at fitness values, it is easier to predict when a catabolic gene is relevant for the growth of the strain because it usually has a clear, clean phenotype. But when you are looking at these signaling components, there's a plethora of effects that could be happening within the cell, and it's a little bit more complicated to distinguish or discern which is actually relevant for the sensing. So to address that, what we did is that we ran a co-fitness analysis that what it does is that it clusters genes together that have a similar fitness profile. And that you can see here in this heat map. In the y-axis, what you can see is different carbon sources. So again, this is in minimal media only containing these carbon sources. And then in the x-axis, what you can see are all of these genes with the annotative functions that are clustered together. Some of the things that started like popping up and that were like a good lead to start were, of course, this 2675, the PET-F, which is what Mitch Thompson et al. Already shown before. And then we could see like some alcohol dehydrogenases. We could see like, as you can see here, the 2682, or we can have like, we were able to see like some hybrid histidine kinases, another one over here. And the first thing that we need for our biosensor is a sequence that is actually able to respond to that analyte. And that's what you can see over here. We decided to just first test the upstream sequence of 2675. And that's what you can see here. And we just coupled that with the fluorescence marker, which is mCherry. And then we were asking how would the expression of mCherry change in the presence and in the absence of isoprene. And that's what you can see in this graph next to this diagram, in which we are looking at fold mCherry induction, which means how much mCherry is being expressed in the presence of isoprene versus in the absence, as a basal expression just to normalize that. And indeed, we were able to see that PEDF was actually responding to Isoprenol, which was a great hit. Something that was very, very crucial for our pipeline, and just like to highlight the need of this step, is doing an RBS optimization. The RBS, what it would do is that it would help you optimize the dose response of the biosensor to the different concentrations of your inducer analyte. And that you can see here, if you look at this red line, which is the optimized RBS, compared to the blue line, which is without the optimization. You can see that not only the linear dynamic rates improve, but also like the fault response, the total fault response versus lower versus the background. And now that we have a biosensor that actually responds to isoprene, we can interrogate all of the rest of the candidate genes to see which ones are crucial for that response. Because if you look at this diagram here, if we remove some of these components, then isoprene wouldn't be able to induce the biosensor. And that's what I'm showing here in this first bar chart, in which, so control would be like the wild type strain, and this is like the fold and chain induction. And then like we just generated simple, clean knockouts. Something to see, for example, like I just want to point out 2665, which is a response regulator. And as you can see, the fault chains, like the response to the sensor was completely blocked, meaning that that is, or highlighting or pointing out that that is the response regulator responsible in this signaling cascade. And another interesting thing is to look at this YIAZ, which is 2682. This is a histidine kinase. 2683, my bad, a histidine kinase. I want to now move to the next bar chart here in purple and in yellow. This is a histidine kinase, and this is an alcohol dehydrogenase. The deletion of both blocks the response of the biosensor. And then the next logical step to actually be able to link this genotype to the phenotype that we're observing is to complement the function by reincorporating these genes into the mutant strains. And then like this is where we found one of the first interesting things in this research. That is that the complementation of in this, for example, histone kinase knockout, the complementation of the histone kinase was not enough to restore the activity, but it was when both the histone kinase and the alcohol dehydrogenase were co-expressed together, as you can see in this section over here. And the same happened for the alcohol dehydrogenase, highlighting that there must be an intrinsic or a complex interplay between these two modular elements, and that the stoichiometric levels of both of them were actually playing an important role in the signaling cascade. And we will see later on that it actually was the case. And then since we currently don't have a crystal structure of these proteins, but we are currently working on getting that crystal structure, we decided to resort to AlphaFold multimer analysis to be able, if we could predict which would be the interaction domain between the alcohol dehydrogenase and the histidine kinase. It's remarkable to note that alcohol dehydrogenases in bacteria, like they play a plethora of functions apart from just being alcohol dehydrogenase or its canonical role. But it was not described whether they could have this modular necessity in a signaling cascade like this. And so going back to the point, what AlphaFold multimer analysis predicted is that there was a 14 amino acid domain in the N-terminal of the alcohol dehydrogenase that was interacting with the N-terminal of the histidine kinase. And when we made that knockout, is what you can see here, that was enough to prevent the induction of the biosensor. Highlighting that, yes, they do need to interact with each other for this signaling cascade to actually work. And I'm not showing this data, but we also confirm this by other like biochemical assays, especially like co-expressing them in E. coli and then doing a gel filtration analysis. And both of them were coming up together, suggesting that, yeah, or like showing, or we were able to observe that they were actually forming a strong interaction. And then something else that I found like truly interesting is that we also use AlphaFold 3 ligand analysis, and just to try to predict what would be the active site of this alcohol dehydrogenase. And we were able to see that this histidine 281 was playing a crucial role in binding to the cofactor NAD+. And when we mutated this residue and we changed it for an alanine, we observed that that was enough to block the response of the biosensor or of the signaling cascade, highlighting that the alcohol dehydrogenase is not only playing a role as a structural component, but its activity is also important for this signaling system. And something else that I wanted to show you is that how important these findings were for us to improve the activity of the biosensor. Since, so here we have just a schematic representation of this signaling cascade in yellow and purple, this histidine kinase and alcohol dehydrogenase, which both are needed to activate the response regulator, which in turn binds to DNA, driving the expression of the downstream genes, in our case, mTERI. And here in the plot that I'm showing next to it, again, this is like full mTERI induction. I want you to look at the blue line over here. This is when we constitutively express these modular components in the strain. We were able to see a huge increase in the, or improvement in the biosensor dynamic range. And that we was also able to observe that using flow cytometry, in which in the first panel we are looking at the control strain, and then in the second panel the strain with this constitutive expression. And what you can see in the control strain is that we start to see, I don't know if you can clearly see that, but we start seeing like a bimodal population at the 10 hours after induction, meaning that this bimodal population is one part is non-induced and another part is induced. And at the 24 hours, we're seeing like this double population. However, when we look at this constitutive expression, we're starting seeing this activation of the biosensor at the 2 hours post-induction. As you can see here, not 2 hours, no, my bad, at the 4 hour, the bimodal population starts to appear. And then by 24 hours, practically all the cells are already mTERI positive. And moving forward, there's something that I skipped, but that I wanted to also bring to this presentation because I also found this like really, really cool. So what I wanted to show you is that we made this knockout of the 2664. This is another histidine kinase that came up in our co-fitness analysis. And what we observed is that it's actually improving the response of the biosensor, no? Which was quite interesting and was highlighting that this second histidine kinase might be having a role in regulating the signaling cascade. And we decided to, I'm sorry, we decided to do an affinity pull down assay and detection by mass spectrometry, in which we tag this histidine kinase, the 2664, with histidine, and then we co-express it with our histidine kinase, our primarily histidine kinase that we were seeing before. And after expressing both enzymes and then passing them through a column with like nickel beads, we were able to see that there was a high or like a higher recovery of YIAZ, highlighting biochemically that these two proteins are actually interacting within each other, no? And forming like a stable complex, forming a heterodimer, which is also not something so conventional for histidine kinases. And then a little bit of the biological aspect of how these two histidine kinases regulate the response for short chain alcohols. I want to show you, or I want to bring this plot, this experiment, because it truly shows the complexity of signaling systems. And is you here again, we are looking at full mTERI induction. In white, we have the control wild type strain harboring the biosensor, meaning that it has both histidine kinases. In red, we made a clean deletion of 2664, meaning it only has YIAZ. And then in purple, we deleted YASC, meaning it only has the 2664 histidine kinase, not this one that we were seeing over here. So the first thing to notice, which is pretty clear, is that when we delete the YIAZ, there was no response of the biosensor for any short chain alcohol. Then, like pointed out, that that's the main histidine kinase responsible for the signaling cascade. And then when we compare the response of the white strain, no, and the red strain, we were able to see like a different response for different alcohols. So starting for isoprene, which is the first bars that we have in this plot, again, we were seeing that the deletion of 2664 was improving our response to the response to isoprene, no? Meaning that it could be like attenuating or reducing the heterodimer would be reducing the signaling for isoprene. However, something very interesting is that we also observe the opposite phenotype, in which having this heterodimer of both the 2664 and the YIAZ gave a better response for ethanol, as you can see here, if you can look at my cursor. Which then it was really interesting to see how these hetero versus homo dimers were attenuating or influencing the response, or how Pseudomonas putida can respond, no? Can modulate the response uh, um, to be able to catabolize all of these short chain alcohols. And then I'm going to move forward into something else, into how we actually wanted to repurpose this biosensor to improve isoprene production, which was one of our main goals in Pseudomonas putida. So to do so and to improve the high throughputness of our assays, we decided to create conditional auxotroph strain. We did this by deleting PYRF. This gene is needed to synthesize uracil, meaning that if you have this knockout strain in minimal media and there's no uracil, the strain cannot grow at all. So if we replace then the M cherry for the expression of PYRF, then that means that isoprene is going to drive the expression of PYRF, and then it's going to rescue the growth of the strain in minimal media, but only when isoprene is present. And to do so, again, I want to highlight the relevance of doing RBS optimization. And this is something that we can see here. Maybe it's clear to look at these plates first, these simple photos, in which we did again an RBS mutagenesis. And what we wanted to do is we wanted to reduce the leakiness expression of PYRF, so that if there's no isoprenol, the strain is not expressing PYRF and it cannot grow. And this is like crucial for this assay. That's something that, for example, you can see here. If we look at this clone, at this mutant here, you can see that there's no growth when there's no isoprenol. Then when we added 300 milligrams of isoprenol, there's partial growth. And then when we added 1 gram per liter, there's saturated growth, like a really good growth. And that, if we look at this plot in the left side of the screen, we are looking at OD values. So the original RBS that we were using, as you can see, when there's no isoprenol, only glucose, this first bar here in yellow, there was like fine growth, like actually saturated growth, meaning that there's a huge leaky expression of PYRF. And then if we look at our two mutants in purple and in blue, you can see then in neither of the cases the strain was able to grow without isoprenol. And then, for example, if they have like different thresholds of astringency. As you can see, the purple one was able to grow with 300 milligrams, but the blue one was only able to grow when 1 gram per liter of isoprenol was present in the media. Again, highlighting this dose response that the RBS is causing. And then to develop this high throughput growth assay, what we decided to do is that we wanted to use a guide RNA library. This works similarly to the transposon library that I was mentioning at the beginning of the presentation, in which we use a guide RNA library with CRISPR system that knocks down these genes, and our guide RNAs were covering practically all open reading frames in Putida, in the genome of Putida. And then what we do is that we grow this auxotroph strain with this guide RNA library, and then what we are asking is that something that I'm sorry that I didn't mention, and I didn't want to get into these details because I wanted to focus the presentation more on the biosensor than in the production aspects of the paper of the research, is that there was already described a pathway to produce isoprenol, and we just integrated that into the genome, and then we use this guide library. So we induce the strain to produce isoprenol. We add this guide RNA library, and then if there's any targets, any knockdown that increases the production of isoprenol, that means that it's going to increase the growth. And that means that that guide RNA at the end of the experiment is going to have a higher abundance because the strain can grow better and it's going to outcompete the other ones. Um, and that's what I'm showing here. We just sequenced the guide RNAs at the end of the experiment, of the cultivation. We did several rounds with those different RBS that I was showing before that has different levels of astringency. And then, as you can see in this plot here, I think it's pretty clear. In the bottom section of this clear, it shows the RNA coverage. As you can see, it's covering most of the, or like practically all the open reading frames in the genome. And then what we can see like in this, like above, is what were the RNAs that were recovered after these growth selection assays. And although knockdown, it is not the same as a knockout, we decided to see like how the knockout of these genes would play a role in isoprene production. And that's what we started doing, thanks to, by the way, Morgan Apollonio, which was an undergrad that was working with me, and she helped me really elaborate all this combinatorial space, which is a huge space. And we just ranked our hits, our guide RNA hits, um, based on the abundance in which they were found at the end of the experiment, and then we started like generating these clean knockouts, and then starting like making like doubles, triples, quadruples, just like trying to expand this. And something that was really interesting is what you can see here. So in the top of this plot, this is like a summary. We started doing like these single knockouts and then going to the two deletions, three deletions, etc. These icons just represent different gene classifications that you can see here in the bottom left of the screen of the plot. And then we use a heat map to represent how much isoprene are what is strains producing as these knockouts were being generated. And something that was actually truly interesting is that most or like all of these knockouts that were improving the production of isoprenol didn't have a clear or an intuitive relationship to isoprene production. No? Just to give you an example, this first one that we made, this first knockout, the 2428, that we already seen like quite a nice increase in production. It's a sugar transporter. Then we have 4622, which is a regulator in tyrosine degradation. And again, I think that highlighted how this RNA library really expands the ability to find these non-intuitive targets, which we wouldn't have been able to find if it was not because of the growth selection assay. And in this bar chart over here, we love this kind of bar charts, but I didn't want to, again, bring that much production values. But I just wanted to show you how these are just like the name of the strains as we were making all of these modifications. And we started with a strain that, well, was actually not a great producer. It was like 50 milligrams per liter. We did some optimizations of the media and like some proteomics to understand its differences based on the format that was used for production. And we already jumped to like 150, 200 milligrams. And then doing all of this combinatorial space, we actually reached like the 900 milligrams per liter, which is a huge increase in a product of this type in a microbial host or in a bacterial host. And then something that I was saying before, as part of the M group, we really love understanding which is the physiology underlying all of these changes, no? Like what happened in the strain so that now it's actually such a huge producer and before it was not, no? What changed? How is the strain reacting to all of this that we're doing and accommodating this burden of having to produce something that is not valuable for it? And that's what I wanted to show you here. Just bear with me because I know that this is like it's pretty crowded, but there's a single message that I wanted to communicate with all of this, which is that, yes, first of all, we did proteomics and metabolomics, and there was a concordance with those results, which is always great. And the key finding that we observed, especially from metabolomics, was that there was an accumulation of amino acid pools in production phase when there's no more carbon substrate for the strain to be converting into a final product, no? An isoprene. So let's look at this metabolic map a little bit more closely together. What I'm showing here is that, well, this is like just a simple representation. Of course, this is like a 2D map, like metabolism is more dynamic and more complex than this. But we generated this with the results that we found from metabolomics. So you can see that under some of these metabolites, there's like these four squares. As you can see here, this is the legend. These are two of our best producer strains, and then we sample in growth phase and in production phase. And what we saw is that if we look at pools of phenylalanine, tyrosine, and leucine, there was a high accumulation of these amino acids in these engineered strains compared to the most, like the less produced strain in production phase. Something that our hypothesis was that the metabolism required to accumulate these amino acids in production phase because isopenol, and I didn't mention this before, is only produced after the growth phase. And something that we did to validate this hypothesis and is that we overexpress tyrosine degradation enzyme. This is the bar chart that you can see here that I'm trying to mark with my mouse. And what we see is that just increasing that enzyme, just increasing the degradation of tyrosine, we were able to see in production phase, we were able to see an increase in isopenol production in the most simple or less edited strain. In the high production, we didn't see that much of a change, um, probably because it's already rewired to do so. And something else that I think is pretty revealing about all of this is that we look at the protein content of different protein categories or like types compared to the total protein abundance. This is this chart that you can see in the top right side of the slide. And what I'm showing here is that we are comparing this strain, which is one of the most, or like one of the less edited strains. So it doesn't produce that much compared to these two best producers. And the first things to notice is that the isoprenyl pathway proteins, the abundance of these proteins labeled with this brownish reddish color, was the same for the three strains, which this is like pretty it's pretty interesting because traditionally, metabolic engineers, we tend to assume that an increased amount or an increased abundance of pathway proteins actually is what is the bottleneck for improving production. But in our case, it wasn't. As you can see, that same abundance of proteins, huge change in production profile. Something as a control that you can see is that it's actually, we were looking at the flagella genes as well. We deleted FLEQ, which is the master regulator for flagella synthesis. And of course, that was then downregulated in our best producer strains. And then while we actually tried to delete the PHA formation, granule formation genes, and we didn't see any increase in production, we observed that our strains had natively rewired through all of this engineering process to reduce the abundance of those proteins. These are carbon sinks known in Pseudomonas putida. And that's what you can see here in purple, that the less produced strain has higher abundance than the our base engineering strains. We can get into more details of these volcano plots. I don't think it's for the purpose of what I wanted to communicate. I don't think that's necessary. It's just like there is a correlation between both of them. We see, like, for example, this ACEA protein we see an overexpression, which is it participates in the glyoxide sound, and that's what we see here in TCA cycle as well. And then like some HMG proteins, which are involved in tyrosine degradation. And this is also something that we deleted the tyrosine degradation regulator. And yes, I hope this slide, I know is very busy. I should break this down in several slides, but I hope it was clear. But we can answer any questions at the end. And this is everything that I wanted to bring up today. Thank you very much, really, for your time and for inviting me here to be able to share my work in this this biosensor seminar series. And of course, I wanted to thanks my research group, the EM group, especially Andrula and Thomas, my PIs, for giving me the opportunity to do such nice research and being able to do a PhD at UC Berkeley. And thank you so much.